Everybody doing okay? Finally getting settled into class. I don't know what time it is. I feel like I know we're past where we should have started already, so I'll jump over to my to-do list as fast as I can. I got here a second ago and realized the breaker was, I guess the storm knocked out all the power in this room, so the coffee maker and everything I had in the fridge was all ruined. So I'm running on fumes right now with no fresh coffee. I normally have that, so we'll see how this goes. And it's going to be necessary for me to have some kind of fuel because we've got 2 PowerPoints to get through today. It's all of the respiratory system stuff. The quiz has already been up on D2L, so hopefully you've been able to mess with that. I've passed out the respiratory lab list. You can see on Thursday all we have is review stuff. For Unit 3, it seems like it's stretched out over spring break, so it's been a long time. Starts with lymphatic system, includes immune system, and then we complete it with respiratory system stuff. The first part is more anatomy based. We'll talk about the functions of the respiratory system and then some of the structures. A lot of those are on the lab list. And then the second part is about gas exchange, and we're going to draw the spirogram. So you have one of those that's already labeled. I'll just draw one of those and talk about it on the board. Before I pull down the slides, I thought we could maybe do a slower version of filling in that picture that I'm drawing, because I think I can answer some of the questions like functions of the respiratory system and, well, just some other basic things that are confusing. I don't know that drawing is not in itself somewhat confusing, so maybe I can start here. And if you just came in, you haven't really missed anything. It took me a while to set this camera up and get going, so we're just now about to start information. If I can remember back to where we left off last time, I guess that was immune system right at the end of right before spring break. I think I had adjusted. The schedule's been messed up by the winter events a couple of times. So I think originally on the case study that you downloaded on D2L, I probably never updated that deadline on it. So I think that still was like March 10th or something, which we're well past that. So I had adjusted, I think March 25th or something like the end of this week. If you haven't done that case study on immune system stuff, we can certainly talk about that today. I'll be glad to step back into immune or lymphatic stuff. In fact, I've got the handouts that we went over for lymphatic and immune over here if you want to consult those. I didn't make copies of the respiratory system handout for everybody, the ones that are on D2L, but I see most people already have those. I do at least have the keys over here, but I should say all of that stuff when we get through lecture. And one of the first couple of slides, if I were to pull down the PowerPoint, looks like this, but I thought I would take the slower version of trying to talk my way through this before we pull down the slides. So that's the plan. Now, is there any questions about anything or stuff where we left off last time, immune system or case study stuff? No? Okay, then I will focus over here on respiratory system, and we'll pull down the slides in a second, but I guess this starts quiz 6 information. And let me explain what we're looking at over here. You know, I always struggle with ways to represent ideas on the board. And so what's going on here is that this side of the picture represents the left side of the heart. I couldn't just draw the left chamber, so I drew the heart, and I labeled it as the left side of the heart. So what we have here, we know that, kind of remembering back to Unit 2 stuff, pulmonary veins are what take, any vein is going to take blood towards the heart. Any artery is taking blood away from the heart. I said arteries aren't always red. Sometimes arteries are blue. An artery is always carrying blood away from the heart. We said veins aren't always blue. Sometimes veins are red, but what's unique about a vein is it's always carrying blood towards the heart. So what's nice about this picture is we've kind of separated our pulmonary circuit on top, where we go from the heart out to the lungs and then back to the heart, from our systemic circuit down at the bottom. This is where that oxygen-rich blood leaves the heart. It goes through some of the body tissues, which I've represented by just a few cells here. And then by the time we get through those systemic capillaries, we'll see a lot of that oxygen is going to be pulled out, and it's going to be replaced by a bunch of carbon dioxide. So we're using blue to represent oxygen-poor blood. I thought we could put some numbers to that so that we can see how this gas exchange takes place. And I started with some weird numbers over here. And this just happens to be on Earth, obviously, is the only thing that we have been able to measure. I don't know, maybe they've measured this on Mars or the Moon. But in Earth's atmosphere, if we were to take all of the gases that make up the atmosphere, most of them are nitrogen. It's like 79% nitrogen, around 20% oxygen. There's a bunch of smaller gases than that. One of them that we'll talk about is carbon dioxide. It makes up less than 1% of the total gas in the atmosphere. If we were to take all of the gases in the atmosphere and line them up in 1 squared millimeter, the pressure of all of those gases would end up pressing down and weighing about 760 millimeters of mercury, which is how they measure atmospheric pressure. This is probably beyond what we really need to know. I'm just trying to explain where they get these numbers from. So if we were to take each of those gases that make up atmospheric gases and look at their percentage of that 760, most of that 760 millimeters of mercury is nitrogen. But if we were to take about 20% of that 760, about 20% of that gas would be oxygen, and that 20% would account for about 160 of those millimeters of pressure. Again, we don't have to get too deep into you're not going to have to calculate this stuff. Carbon dioxide makes up less than 1%. So of the 760 millimeters of mercury, only 0.23 millimeters of mercury are accounted for carbon dioxide. And this is atmospheric gas, so outside air. When we look at the slides, you'll see the large P, and that just stands for partial pressure. There is going to be a partial pressure of oxygen and also a partial pressure of carbon dioxide. Carbon dioxide is much lower, obviously, in outside air. So here's where we're going to get these numbers. Just has to do with how the atmosphere is on Earth. The partial pressure of oxygen in outside air is going to be 160 millimeters of mercury. We won't talk about elevation change because that changes if you're at like sea level or up at the top of Mount Everest. But this is based off sea level. And the partial pressure of carbon dioxide in outside air is less than 1%. Now that's going to change when we get into the lungs. So in my very simplified drawing here, and maybe to make it even simpler, I was about to erase all of these numbers, but I guess I'll let them sit there for a second in case I get any kind of questions about it. But what we can think about in terms of the functions of the lungs, we're going to see in a simple way that oxygen is going to make its way into the lungs, and we'll see that carbon dioxide is going to make its way out. But we can be a little bit more specific about that. We're going to see that each of these gases, oxygen doesn't care about carbon dioxide. It's going to move down its concentration gradient all by itself. It doesn't care about any of these other partial pressures. Carbon dioxide is the same. It doesn't care about the concentration of oxygen at all. So hopefully I've given myself enough room here. We're going to do one of these gases at a time because neither one is dependent on the other. We'll start with oxygen out here in the outside air. We have the partial pressure of oxygen in the atmosphere. It's 160 millimeters of mercury. But what starts to happen when that 160 millimeters of mercury makes its way into the lungs? This is a very simplified lung. When we pull up the slides, we're going to look at all the respiratory structures. We'll split them into upper and lower respiratory tract structures, and at the very base of the lower respiratory tract structures, we'll eventually get to these alveoli, which we're going to call the exchange surfaces of the lungs. We're going to see 3 cells, in fact, that line these alveoli, and we'll have to know the functions of those 3 cells, but I'm getting ahead of myself. Welcome. Glad that you survived the storms last night. That was crazy. There you go. That's just the lab list. So we're starting. I haven't even pulled down any of the slides yet. I was just trying to get this diagram up that explains some of the functions of the respiratory system, and then we're going to catch up with slides. There's a lab list that you've got, but Thursday there's just review questions about lymphatic, immune, or any of the respiratory system stuff, a practice practical, and then a week from today we've got an exam. I just have to say that a couple times because the weather events have messed us up so many times, the schedule's hard to keep track of. So if you just jumped in here, we were talking about the partial pressure of gases in the atmosphere. And on Earth, the outside air has a partial pressure of about 160 millimeters of mercury of oxygen. We talked about that number comes from the fact that 20% of the atmospheric gas is oxygen. Less than 1% is carbon dioxide. So if we put the partial pressure of carbon dioxide out here, you can see it's very low when we're thinking about outside air. But this is where I was about to move forward with the story. So we're going to take one of these gases at a time, and we'll start with oxygen. We're going to see that as soon as the oxygen starts to move inside the lungs, I know I've really shortened the lungs here, we're going to see that the cells are going to start to use that oxygen. So the oxygen levels are going to end up dropping by the time we get all the way into these exchange surfaces. The partial pressure of oxygen inside these exchange surfaces is going to go all the way down to 100 millimeters of mercury. And I'll just go forward, I guess, with this. We're going to see that the partial pressure of oxygen inside the alveoli is going to end up balancing with the partial pressure of oxygen inside these pulmonary capillaries. It's going to be 100 millimeters of mercury. I was about to explain what happens over there, and we're about to get there, but we're going to take a loop around first. So here's this loop. The 100 millimeters of mercury, this amount of oxygen gas that has entered the pulmonary capillaries, is going to stay the same as it goes through the left side of the heart and out of these systemic arteries. So partial pressure of oxygen is still 100 millimeters of mercury. But that's about to change as we go through these systemic capillaries. All the cells down here are basically pulling any of the oxygen that they can into the cell, and they're all creating carbon dioxide that they're pumping back out. As they carry out their cell activities, they're releasing a bunch of this carbon dioxide. So as we move blood through these systemic capillaries, I guess that's the one that I had, we're going to see partial pressure of oxygen by the time we get to the far end has dropped all the way down to 40. And the reason that we've lost 60 millimeters of pressure here for oxygen is that that's been gobbled up by all of our cells that are pulling that oxygen in to do their cell activities. So oxygen levels are going to drop. Those oxygen levels are going to stay really low, still at 40 millimeters of mercury, as they move into these pulmonary capillaries. It's a terrible arrow. Okay, so like I said, we're just concentrating on oxygen all by itself. We've got a low concentration of oxygen being pulled or pushed into these pulmonary capillaries. Oxygen is going to go from an area of high to low concentration, so as blood moves across these pulmonary capillaries, oxygen is being picked up, so that the partial pressure of oxygen is much higher when we get into the pulmonary veins, which is why we're drawing that in a red color. So high concentrations of oxygen, we draw that in red. Low concentrations of oxygen, we draw that in blue, our oxygen poor blood. We've just gotten through oxygen and we're about to pick up carbon dioxide. Both of these pressures are independent of one another. So the story that I'm about to talk about with carbon dioxide has nothing to do with the story that we just went through with oxygen. So this is a new one. And we're going to see that the atmospheric levels of carbon dioxide are less than 1%. They're very low. So the numbers that we were looking at over here is how they measure the pressure of atmospheric gases. If we had all the gases in the atmosphere and we combined how much pressure they can push down, they end up weighing 760 millimeters of mercury. That's all of the gases. We're not concerned with all of the gases. We're just thinking about oxygen and carbon dioxide. So oxygen makes up about 20% of that 760. So out in the atmosphere, oxygen has about 160 millimeters of mercury. When it got into the cells, we saw that number start to decrease because cells are using that oxygen and instead pumping in a bunch of carbon dioxide. So we're about to talk about that carbon dioxide. Out in the atmosphere, carbon dioxide levels are very low, but what starts to happen as soon as we get into the lungs, we're going to see carbon dioxide levels are increasing because all of these cells have been pumping out carbon dioxide. We'll put the partial pressure of carbon dioxide in here. Hopefully I've given myself just enough room. Partial pressure of carbon dioxide in here is going to be 40 partial pressure of oxygen and partial pressure of carbon dioxide is going to reach equilibrium on either side of the alveoli and pulmonary capillaries. This is what we're going to call external respiration, gas exchange between outside air and the bloodstream. This down here is going to be internal respiration, where we have gas exchange between blood and body cells. One of the reasons I'm going through the trouble of drawing this is so that I can separate external respiration from internal respiration. So this bit is external respiration. We're going to see carbon dioxide levels. So we're going to see, again, outside air and pulmonary capillary partial pressures of gases are going to balance themselves. Just a long way of saying that partial pressure of carbon dioxide as we move into the pulmonary veins is also 40. Matches what it is inside the alveoli. And that stays the same as we go all the way through the lungs. We can see that partial pressure of carbon dioxide over here is still 40, same as it was in the pulmonary veins. But you know, as we go through here, carbon dioxide is just being pumped into the systemic capillary. It's being picked up by the bloodstream. So by the time we get through the systemic capillaries, the partial pressure of carbon dioxide is going to be higher than 40. It's not a lot higher. It's only up to 46 millimeters of mercury. But again, that's higher than it was. So now this blood that is high in carbon dioxide and low in oxygen is going to make its way through the right side of the heart. The pressures are going to stay the same until we hit those pulmonary capillaries. So partial pressure of carbon dioxide is still at 46 millimeters of mercury until, again, we move through the pulmonary capillaries. We're going to see carbon dioxide diffuse out. So there goes that carbon dioxide out, and at the same time, this blood is low in oxygen, so it's going to be pulling oxygen in. If you remember from 1406 recently, gas is like carbon dioxide and oxygen. They're tiny. They're non-polar. They don't need any kind of special gate to move across a cell. They can just drift freely across that plasma membrane. So what determines the direction that they move is their concentration gradients. We can see oxygen is constantly moving down its concentration gradient. So is carbon dioxide. It's moving from high to lower concentration and then eventually into outside air. Hopefully the numbers aren't confusing, but what I was really trying to separate there is this thing that's happening right there, this gas exchange between outside air and the bloodstream. This is what we're calling external respiration. Gas exchange between outside air and the bloodstream. And then this bit down here is internal respiration. That's gas exchange between the bloodstream and body tissues. What else? There's a couple of other things that we need to talk about that take us into the second part of the PowerPoint. The second part of the PowerPoint there is gas exchange. So we need to talk about, I'll just put it in here in the middle as gas transport. What I've left off of this drawing is how is it that oxygen travels through the bloodstream? How is it that carbon dioxide, what is it that carries those things through the bloodstream? We'll answer that with the gas transport part. And then the other part there, the spirogram, you have the last part of that lab sheet, you have a spirogram on there. And that is graphing the movement of air to and from those exchange surfaces. It's measuring what we're going to call pulmonary ventilation. So we'll talk about pulmonary ventilation, this movement of air to and from the exchange surfaces. We'll talk about the muscles involved in inspiration and expiration. But at least we can see one of the main functions, which is gas exchange, and we can be specific between external and internal respiration now. Pulmonary ventilation, gas transport, that's where we're going. Everybody still doing okay? All right, let's get into some of these slides. So here we go. Jumping into the first slide, you can see we're going to go through the functions of the respiratory system. And it's kind of too simple just to say providing oxygen and getting rid of carbon dioxide. We know oxygen comes in and carbon dioxide goes out, but we wanted to separate what we're going to call external respiration, like we said, gas exchange between the bloodstream and outside air, from what we are calling internal respiration, this gas exchange between the bloodstream and body tissues. So somewhere else in there, we're going to talk about our pulmonary ventilation and gas transport. But a couple of other functions that we can list for the respiratory system. One of them is speech, what I'm using to communicate with everybody now. And then the other one is smell. So we saw with the large half-head model, we talked about the nasal cavity, being able to pull in air very quickly to the superior part of that nasal cavity so that it comes into contact with olfactory epithelium and you can smell if somebody's giving you a flower. Or like in the case for my dogs, I'll always like throw a bunch of milk bones somewhere and then they go smell around, see if they can find them. Here are the four that we need to be aware of. Pulmonary ventilation, we abbreviated that as PV, but that you can describe as the movement of air to and away from these exchange surfaces. We're just going to talk about inspiration, take an in breath, and expiration, just exhaling. So inhaling and exhaling, getting air to those exchange surfaces is our pulmonary ventilation. That's what we graph with that spirogram. Then we've already talked about external respiration. We know that's gas exchange between outside air in the blood versus our internal respiration. And I said somewhere in the middle there's the transport of gases, which we cover how carbon dioxide and how oxygen move through the bloodstream. Hint, it has something to do with a red blood cell. But this is what we just drew on the board, and I'm glad to finally be able to do this because last semester I didn't get to draw that on the board. We had to do this in a series of pictures because I was just stuck at home talking through those slides. And on a side note, I think I did remember, it's been a long week, but I think I remembered to put those things back up. So there is a slower version of me talking through this stuff, and that's why I had to have those boxes. So I was wanting us to focus on the highlighted portion in here where we're thinking about atmospheric air, has a higher concentration of, in fact, if we just look at oxygen by itself, it's at 160 millimeters of mercury at outside air. This is where we get those partial pressures of gases. So I'll jump over that real quick. But we see oxygen goes from 160 millimeters of mercury down to 100 when it enters the lung. Again, cells are already starting to use up some of that oxygen. It stays at 100 millimeters of mercury all the way through the left side of the heart until we get into body tissues. And then it's going to start to drop as those cells use oxygen. It goes from 100 millimeters of mercury down to 40 millimeters of mercury as the cells have depleted that oxygen. And it returns to the lungs at 40 millimeters of mercury. And as it moves through those pulmonary capillaries, we see that oxygen moves down its concentration gradient. It once again absorbs enough oxygen so that it's right back up to the 100 millimeters of mercury. So we pick up oxygen in the pulmonary capillaries, and what we're going to get rid of is the carbon dioxide. You can see carbon dioxide is very low out there in atmospheric air, but as soon as we get into body tissues, that partial pressure of carbon dioxide, there it is at 40, has drastically increased. Again, because cells are dumping that carbon dioxide out into the extracellular fluid. As soon as we get into the lungs, as soon as we get into the pulmonary capillaries, that partial pressure of carbon dioxide is still at 40 millimeters of mercury. It's not until we pass through those systemic capillaries that it picks up that additional carbon dioxide. It goes from 40 millimeters of mercury up to 46, which isn't a whole lot, but it's higher than it was, and this 46 millimeters of mercury stays the same till we enter the lungs, and it's only 40 inside the alveoli, and it's 46 in the pulmonary arteries. So carbon dioxide is going to leave the pulmonary capillaries, going down its concentration gradient. So we can be a little bit more specific about what happens during this external respiration. Oxygen moves into the bloodstream and carbon dioxide moves out. Which is, I guess I just said all of that with my little pointer here. This again is just showing oxygen levels are at 100 millimeters of mercury, just like they are when they enter the systemic capillary. But at the end of the systemic capillary, they drop down to 40. They stay at 40 until we get back to the lungs. Okay. We drew that, so hopefully that was helpful. The next few slides take us into some of the structures of the respiratory system, and I just put this dotted line in there that it lands right there on the larynx, or the voice box, and that is the structure that we use to distinguish upper respiratory system or upper respiratory tract structures from lower respiratory tract structures. Okay, so there's that. The other thing, and this is one of the lecture handout questions, it wants to know the difference between the conducting zone and the respiratory zone. The conducting zone is going to be any of those passageways that end up doing things like filtering the air of any debris, or they might have to warm or cool the air before it hits those exchange surfaces. You know, sometimes if you get into, always like when the weather changes, like if you go outside and it's really cold, if the air is too cold, your body has to warm that air before it hits those exchange surfaces. Or if you get into like a car that's been sitting out in the parking lot in the sun for too long, sometimes that air is real hot in there and it's hard to breathe if you take in that hot air. So the air needs to be cooled sometimes. It has to be at the proper temperature and the proper humidity before it hits those exchange surfaces in order for that gas exchange to take place. So the conducting zone, these are all going to be respiratory structures that don't have anything to do with gas exchange. They just have to do with preparing that air to hit the exchange surfaces, making sure it's free of debris. The respiratory zone, what's different about the respiratory zone versus the conducting zone is that you know that you're in the respiratory zone because you see alveoli. Those little pockets are where gas exchange takes place. If gas exchange is occurring, then we're in the respiratory zone. So we don't enter the respiratory zone until we start to see these alveoli. Those are the dead giveaway. So actual sites of gas exchange, we're going to see there are going to be what we call respiratory bronchioles that have alveoli, and this is where the respiratory zone starts. This is also going to go down the lab list as well. I would probably use this large half-head model to point to some of these structures. There is the external nares, which you can just think of as the nostrils. So air is going to enter these external nares, and now is in this, at first it's in this area called the vestibule. And then while it's in this vestibule, it could enter through either one of these air passageways that we called the meatuses. There's an inferior nasal meatus, a middle nasal meatus, and a superior nasal meatus. Then those little shelves that stick off the side, kind of the wall of that nasal cavity, those shelves we called nasal concha. So there's a superior, a middle, and an inferior nasal concha. If we go, let's say we went through this middle nasal meatus, where a little a piece of air that made its way through the middle nasal meatus. When we emerge through the other side of that nasal meatus, we're in this area called the internal nares. It's the very back portion of that nasal cavity. After we cross that internal nares, we're really in the back of the throat. What else do we want to say here? I'm going to jump over that slide just for a second because I was just talking about as we cross over this imaginary line that we refer to as the internal nares, kind of the last bit of that nasal cavity. We then enter what we consider the throat or the pharynx, and this is going to be broken into three different segments. There's the part of the throat that's shared with the nasal cavity. We call that the nasopharynx. We might have seen this earlier in the semester too. There's the part of the throat that's shared with the oral cavity. We call that the oropharynx. And then there's the last part of the throat that's shared with the larynx or the voice box. We call that the laryngopharynx. So three parts to the throat or three parts to the pharynx. If we go all the way, so what's interesting is that all these parts of the pharynx are going to be covered in stratified squamous epithelial tissue, just like the surface of your skin. But as we start to enter this respiratory tree, we're going to start to see the layers of epithelial tissue change. And we don't have to do this yet, but it's interesting. We start to see that the layers of epithelial tissue are going to change from stratified squamous, so a bunch of these little flat pancake-shaped cells, to as soon as we get into the trachea, and that's the slide that I just skipped over. If we could have gone back to this slide, you can see there are cells that are showing you pseudostratified ciliated columnar epithelial tissue. That is as soon as we enter the trachea. We're going to see stratified squamous epithelial tissue is going to change into this, well, I don't think I'm going to be great at drawing these things, but I'll just put some nuclei in there and some cilia that represent, we've got pseudostratified ciliated columnar epithelial tissue. That's going to give way to just regular old columnar epithelial tissue once we get into some of the larger bronchioles. And then these columnar-shaped cells are going to turn into cube-shaped cells. And then so simple cuboidal epithelial tissue, we'll see it go from simple cuboidal eventually into those alveoli, those exchange surfaces, where it's down to simple squamous epithelial tissue. And the whole idea here is that we're getting thinner and thinner cells to where it's eventually the thinnest type of cell and gas exchange is occurring. So there's a trend and a change in epithelial tissue when we go through the respiratory tract. I saw pseudostratified ciliated epithelial tissue, and that tells me that we're in the trachea or some of those larger primary bronchi. Okay. So let's get past the pharynx and into the larynx. We've got a couple of models over here that show us the larynx, and they each have their good points about them. One of them, they have muscles on it, and obviously this one they have removed all of the muscles. But we can see the larynx from the side view. There are things on the larynx that we need to be aware of, like this flap of cartilage that sits on top of the larynx. We call that the epiglottis. It looks like this big tongue thing that's moving around on this model. And what it's supposed to do when you swallow, this epiglottis is going to come down and prevent things from entering this trachea, from going through the glottis and ending up in this windpipe. So that's also something that I could point out right here. I don't know, I guess I won't reveal all the crazy misconceptions I had as a kid, but it's interesting to think there's two tubes that are right there. You've got a trachea that's just anterior to the esophagus. So if you put you can actually feel those ridges of the trachea, those cartilaginous rings that are on the anterior part of that trachea. And just behind that is going to sit the esophagus. It's worth mentioning because if you swallow a really large food item, sometimes that can end up taking some of the space of the trachea, choking somebody, and you have to get behind and give them the Heimlich maneuver and clear that air passageway. Anyway, we're still talking about the larynx. This is the larynx looking at it face on. I believe the lab list has pieces of cartilage on it that we have to know. This large, broad piece of cartilage that we call the thyroid cartilage, that's where you would find the thyroid gland. There is the thyroid gland. It sits right on top of that thyroid cartilage. Beneath the thyroid cartilage, there's this, it's also a large piece of cartilage, not as large as the thyroid cartilage, but still larger than just the regular tracheal rings. This is what we call the cricoid cartilage. So thyroid cartilage, cricoid cartilage. And then if we can turn this model around to the back side, you can see two other pieces of cartilage here. Bless you. There is, and you can actually grab onto them right here. There are these larger pieces of cartilage that they kind of have this little L shape to them. This is called the arytenoid cartilage. And then at the very tip of that arytenoid cartilage, right there, there's a little piece of cartilage that looks like a tooth. It's called the corniculate cartilage. You can see that on this model as these little tips on top of the arytenoid cartilage. Thyroid cartilage, cricoid cartilage, tracheal rings. We've got arytenoid cartilage, and then the tips there are the, what did I just try and say? Corniculate cartilage. I almost said cuneiform cartilage, but cuneiform is not on the list. Corniculate cartilage, that little tooth-shaped piece of cartilage, is on the list. Okay, what you can also see from here is that those C-shaped tracheal rings, they don't make there's not a complete circle, it's just a C shape, so that the back end of that trachea is actually completed by a little stretch of smooth muscle. In fact, I'm just going to jump over to this slide right here because it shows that C-shaped tracheal ring, and the back edge is connected by this piece of smooth muscle. That muscle is the trachealis muscle. I feel like it's in the lecture handout somewhere. And I said the right behind that trachealis muscle sits the esophagus. So if you happen to swallow a large food item, that trachealis muscle can relax and actually let you use some of the space of the trachea to get that large food item down. It just so happens sometimes if the food item is too large, it could completely block that trachea, and then you've got somebody that's choking and needs to have that cleared. So these C-shaped tracheal rings on the trachea don't go all the way around. What closes that back part of the trachea is the trachealis muscle. Okay, so now I'll go back to where I had just left off. Here we're still talking about the larynx, and there's this little slit-like passageway that's going to let air go down into the trachea. If we could, from the side view it's kind of hard to tell, but if we could go to this view where we're kind of looking above, we're standing on above this larynx model and we're looking down into the larynx model itself. And what we're seeing here, these on the model, I have drawn them in a little bit of a red color. So hopefully if you look on there, you can see the red marker that I used to draw on either side of these vocal folds. What directly touches this little, let me just go to this slide. How about this? That opening right there is called the glottis. That's the opening in which air would drop down into the trachea. But this glottis, this opening, is flanked on either side by these white structures that we're going to call true vocal cords. These true vocal cords on this model was what I was just showing are drawn in red. But on this slide, they're showing the true vocal cords in white. So those directly touch the glottis. Those are the things that vibrate that you use to make sound and communicate with people. But the true vocal cords, you can see that those are flanked on either side. On this slide, they're using a red tissue to show this stuff. These are what we're going to call false vocal folds. So true vocal folds are the vocal cords. False vocal cords are on either side of those. On this model, those false vocal cords are just kind of a tan color. Okay, true vocal cords, false vocal cords. We've got the slit-like passageway itself, which is the glottis. You can see from here those arytenoid cartilages are what open this glottis. You can see those corniculate pieces of cartilage right there. They look like some of that candy corn or whatever. Back to the trachea. So I guess what we're about to do here is go down the trachea and into the lungs. We know there's just one trachea. We can see it from the side view there. Here it is looking a lot like it does on our respiratory tree. I feel like there was the other one that was around here. This one is obviously more colorful. Maybe those are the only two that we have. So we've seen that a couple of times before, and if I'm going to label anything on it, I could potentially label this single trachea. We know that there are primary bronchi that go into the left or the right lung. Once they enter the lung, they continue to branch. So we've seen that at least a couple of times on these models. I've labeled trachea. We've labeled primary bronchus, secondary bronchus, where they branch a second time into the lungs. And we know each of these different colors on this respiratory tree represents a third time that they branch, what we're going to call the tertiary bronchi. So we have to know primary, secondary, and tertiary bronchi. They do continue to branch more than that, which is what this is showing us. We just have to know the first three: primary, secondary, and tertiary. The secondary bronchi are sometimes called lobar bronchi. I rarely use that term, but I might slip up and use lobar bronchi. Segmental bronchi just refer to the tertiary bronchi. Typically, I just use primary, secondary, and tertiary. But they continue to branch like 23 or 4 times more than that, and they keep getting tinier and tinier, where eventually these bronchial tubes are so thin that they're lined by either simple cuboidal-shaped cells or simple squamous-shaped cells. So there's a couple of trends that we're going to think about when we get into this respiratory tree. And one of them I've already talked about with this epithelium, how it goes from originally like in the oral cavity and then the back of the throat, this stratified squamous. Then when you get into the trachea and some of the primary bronchi, it's pseudostratified ciliated columnar epithelial tissue. And then when you get into some of the smaller bronchi, it turns into simple columnar, then simple cuboidal, and eventually in some of the smallest, what am I trying to say, bronchial tubes, where you finally hit the exchange surfaces in the alveoli, you get to the simple squamous epithelial tissue. The other two trends that I don't have on here, but we're seeing now for the first time on the slides, is that there is a trend of decreasing amounts of cartilage. You'll notice if we grab this thing, there are these C-shaped rings along the trachea. And then when we get into the primary bronchi, there still are these C-shaped rings that go into the primary bronchi. But after that, we see that these C-shaped rings will give way to these irregular plates. Let me see if I can get a picture of this. I don't know that this is the best picture, but if we just think about the cartilage itself, there are C-shaped rings in the primary bronchi, and then those C-shaped rings give way to these irregular plates of cartilage. And eventually, when we get to these smaller bronchioles, there's no cartilage at all. Instead, all that is there is smooth muscle. So as we go into the lungs, cartilage decreases, but what increases is that smooth muscle. You'll see at the very beginning, like in the trachea and in the primary bronchi, there's very little smooth muscle. But as we get towards the end of the respiratory tree, there's an increasing amount of smooth muscle. Where you can see at the terminal bronchiole, there's no cartilage at all, there's only smooth muscle. And it's that smooth muscle that can constrict or dilate and determine how much air can make it to those exchange surfaces. Let me go back a couple of slides. So here were those trends. We said cartilage starts to go from those C-shaped rings to irregular plates, and then eventually no cartilage in those terminal bronchioles, where smooth muscle increases when we get further into those bronchial tubes. Okay, so we've said that. Let's catch up. Something that we see towards the end, and I'll just point to it right now, there is a difference between the left lung and the right lung. You can see the left lung over here has a little notch that is carved out of it due to the fact that the left side of the heart is larger than the right side of the heart. That big left wall of the heart causes this indention that we're going to call the cardiac notch. It's on the left lung. But in addition to that cardiac notch, you can see that it also makes this left lung be split into just two lobes. There's this superior lobe of the left lung and then the inferior lobe of the left lung. That's not the same for the right lung. There's three lobes in the right lung, so we have a superior, a middle, and an inferior lobe. That might be obvious, and I know they're not labeled here, but they're going to be labeled on another slide. You can see how there are these fissures in between the lobes of the lung. There are these fissures that are at an oblique angle. So there's an oblique fissure of the left lung, and there is the oblique fissure of the right lung. This right lung also has a horizontal fissure. There is no horizontal fissure in the left lung because there's only two lobes. So just some differences between left and right lung that we can see while we're there. When we jump into the lungs themselves, we can see eventually those smaller bronchioles where the cartilage is gone and smooth muscle is the only thing that's there. It can constrict or dilate and determine how much air flows into the respiratory bronchioles. If you're keeping track on when we hit the respiratory zone versus the conducting zone, we've landed as soon as we hit to respiratory bronchioles. What makes it a respiratory bronchiole is that thing right there. Those are the alveoli, those little pockets where gas exchange is occurring. So now this is the last of the conducting zone, and we're now at the beginning of the respiratory zone. So let's first look at the outside of these clusters of alveoli. We call the entire cluster an alveolar sac. You can think of it as a cluster of grapes, where each individual grape would be the alveoli. In the middle of this, if this was a cluster of grapes, there would be an air passageway that goes right through the middle of those cluster of grapes, and that passageway is what we call the alveolar duct. I think I have a picture of it. Well, you can see a little bit of that alveolar duct here, this hollow passageway that goes to the center of that cluster of grapes. I think there's another picture of it here that shows that alveolar duct that enters the center of these cluster of grapes. Again, each grape is an alveoli. You can see to the outside of the alveoli, those yellow structures are elastic fibers. And they allow these alveoli to expand. They're like those balloons. I don't know. As a little kid, there used to be these balloons that you would get that were tied to like a rubber band, and you would blow it up real big and then just bounce it back and forth on your palm. Maybe I had a crazy childhood. But those balloons, they're really big and they're easy to blow up. They stretch really easy. The balloons that stretch really easy like that, they have a lot of elastic fibers in them. The alveoli start out like that, where they're real stretchy. When they fill up with air, they expand. That's why they used balloons to show this thing. We'll see this when we get to the second part of the respiratory system. Those balloons are supposed to represent alveoli, so that when we pull air into the lungs, they should easily expand and fill up. The reason I'm pointing that out is because if you end up, like there are people that deal with a lot of COPD, this chronic obstructive pulmonary disorder. One form of that can be emphysema, where you lose the elastic stretchiness around the outside of these alveoli. Instead of blowing up a big balloon like I talked about, it'd be like trying to blow up like a plastic grocery sack. It doesn't stretch at all. So that would be, this measure of how stretchy an alveoli is called compliance. So if you have a very stretchy alveoli, it's highly compliant, versus somebody that has emphysema that can't really get as much air to those exchange surfaces because their pockets of alveoli don't expand. They've lost their compliance. Let me go back a couple of slides to see if I left anything out. We can see these clusters of alveoli that we call alveolar sacs. We can see those elastic fibers to the outside of the alveolar sacs that allow them to expand. What we also see on this picture is that just to the outside of the alveoli, there are extensive pulmonary capillaries. That's this part of the drawing that we did. All of those pulmonary capillaries just to the outside of the alveoli are waiting to pick up that oxygen and get rid of that carbon dioxide. So don't be fooled. Remember the red one here, this one would be what? It's not labeled, is it? I don't think it is. This red one, it is a vein, that's right. So remember, blue and red just tell us if it's oxygen rich or oxygen poor, not really whether it's an artery or vein. So the blue in here, these are pulmonary arteries taking this oxygen poor blood to those exchange surfaces, and then it's coming away with oxygen rich blood going back towards the heart. So red ones are the pulmonary veins, blue ones are the pulmonary arteries. We saw this already. What I think is kind of interesting is that if you were to take the exchange surfaces of both of your lungs and stretch them out on the floor or something like that, it's about 1,500 square feet, and that is about the size of a tennis court with the doubles, and counting the doubles, whatever that is, size tennis court. So that's a pretty large amount of surface area that this outside air can just hit. And that's one of the reasons, here we are looking at our most detailed picture yet of an individual alveoli. This is the picture that allows us to see those three different types of cells that we find in the alveoli. And I'll just go ahead and get to the one that I was about to say first. This roaming alveolar macrophage, that little fuzzy one that we see, we mentioned that last time in the immune system discussion. We said that alveolar macrophage was one of our external defenses. And the reason it's considered an external defense, we said, because it's exposed to outside air. If there is some dust or debris on the air that you breathe that happens to make its way all the way down to the exchange surface, I had talked about sometimes like I laugh or will end up inhaling like a piece of food and always wonder like what happens to that piece of food? Does it just block my exchange surfaces? Well, if debris does get down to those exchange surfaces, there's at least one type of cell there that clears, it's a macrophage. It engulfs any of that debris and breaks it down. Cleans up the surface of the alveoli. I feel like I'm going to need to write this down somewhere, so I bet I can use the space that's, we might have enough space right there as it is. So when we're thinking of the three cells of the alveoli, if this is any better, not really. Three alveolar cells, they are, we can see all of them right there. The first two don't get a very descriptive name. They are called type 1 cells. Okay, I do need to erase some of that stuff. Type 1 cells are the most common of any of the cells that you see. All of these tan-colored cells with the purple nucleus, those are type 1 alveolar cells. You can see they're flat, squamous-shaped cells. They're like little pancakes. What we need to know about those type 1 alveolar cells is that those are the site of gas exchange. They're the site of gas exchange. And if we zoom into one of those cells right there, that is one of those type 1 alveolar cells. You can see it's flat, squamous shaped. What we remember about epithelial tissue is that it is always anchored down to some type of basement membrane. So right there in the middle there is the basement membrane. And on the other side of that basement membrane is the capillary wall. Capillaries are made out of a single layer of epithelial cells. And these epithelial cells are sharing the same basement membrane. So this little sandwich here of capillary wall cell and alveolar cell with the basement membrane kind of being the peanut butter in the middle, this is what we call a respiratory membrane. It's made out of, again, a type 1 alveolar cell, a capillary wall, and in the middle of that basement membrane that they share. So it's easy for carbon dioxide to jump into the alveoli and for oxygen to jump into the bloodstream. So type 1 cells are where gas exchange occurs. There is another cell in there, which is the green fuzzy cell. That's going to be what we call a type 2 alveolar cell. These type 2 cells, what they do, they're not the site of gas exchange. In fact, there's not very many of them at all. What they do instead is they produce a substance called surfactant. They are surfactant secreting cells. What this surfactant does is it prevents water molecules from wanting to form bonds with one another. Again, this takes me back to 1406 when we were talking about how each water molecule is like a tiny little magnet and it wants to form hydrogen bonds with other water molecules. Has anybody ever done that experiment where you take like a penny and you just see how many drops of water you can put on top of a penny before the water falls over the side? You see that those water molecules, you can put a lot of drops of water on top of a penny and it forms this little dome of water before it ever falls over the side. Each water molecule is cohesive with one another. It wants to form these hydrogen bonds. And if you think about it, if we think about the fact that this alveoli is lined with humid air, there's some water in there, and those water molecules would want to hydrogen bond with one another, and that would collapse this alveoli. That would not be good for gas exchange. We want as much surface area as possible. So what the function of this type 2 cell is, by producing surfactant, what that surfactant does is it prevents water molecules from wanting to form those bonds. It basically is a lipid-based substance that coats these alveoli, and like we said, it increases pulmonary compliance by expanding each individual alveoli. It prevents that alveoli from wanting to collapse. These two cells go hand in hand. Type 1 cells do the gas exchange as long as there's sufficient surface area. Type 2 cells make sure there's sufficient surface area. And then the last one, you think it would be called a type 3 alveolar cell, but instead they call it an alveolar macrophage. That's right. This alveolar macrophage just clears the surface of any kind of debris. I'll go back to this slide there. So there are those alveolar macrophages just roaming the surface of the alveolus. They're not inside the bloodstream, so they're still considered an external defense when we were thinking about the immune system. Okay, so we've seen our trends as we get into the respiratory tree all the way down to the alveoli. We've listed our 3 cells of the alveolus. We've talked about surfactant. I mentioned this term compliance and how that refers to the effort that you need to stretch out the lungs, that poorly compliant lungs are ones that have lost their stretchiness. And I forget that something like, just sitting here talking to you, or as you're sitting there taking notes and listening to me, it takes somewhere around 3% of your body energy to just breathe, to do this gas exchange, to get air to and from those exchange surfaces. People that have poorly compliant lungs, like people that suffer from emphysema, can sometimes have to spend as much as 20 to 25% of their body energy just breathing, just getting air to and from those exchange surfaces. So I remember I had a grandmother that had emphysema really bad, and we would have to put like chairs between the bedroom and the bathroom because she would get out of breath just going to the bathroom and have to sit and rest but it was taking so much more of her energy to breathe just because of the poorly compliant lungs. Okay, we've seen that already. So this is one of the last two slides of the first part of the PowerPoint, and this is where we're looking at those lung structures. And I think I've already pointed this out when we had a slightly larger picture of the lungs to look at. Remember, the left lung has only the two lobes, the superior and inferior lobe. There is that oblique fissure of the left lung. The right lung has three lobes, so it has an oblique fissure and also a horizontal fissure of the right lung. Again, three lobes, superior, middle, and inferior lobe. That left lung has that little indention that we call the cardiac notch. So there's that. I guess what I want to say about this, because I think that's one of the last two slides before we switch gears. Where we're about to go into this next PowerPoint is we're going to talk about gas exchange. We're going to talk about how we get air into those lungs and out of the lungs. And it's almost going to be like playing these lungs like an accordion. We're going to see that there is this space inside the lungs for, let me try that again. There's space inside this thoracic cavity for these lungs to expand into. And that little space is what we're calling this pleural cavity. We remember talking about the pleural membranes. There's the parietal pleural, which is just to the inside of the rib cage. And then there's the visceral pleural, which is on the surface of the lung itself. And there's a little bit of a gap between those two. So as I take in a deep breath, like that, my lungs are expanding into this interpleural space. And whenever I compress my lungs, we're going to see that it's not too much unlike playing an accordion. I know that maybe using my hands is not as great, but that's what this thing is supposed to represent, that if I were to, we have two balloons in here that represent the lungs, or you can think of them as just individual alveoli. There's a little bit of a hole here at the top, and then there's this rubber membrane at the bottom that's supposed to represent the diaphragm. And we're going to see, as I jump into this next PowerPoint slide, that how we draw air into the lungs is by dropping that diaphragm down. The diaphragm drops down, and we're going to see that the ribs, the muscles in between the ribs, are going to pull the rib cage out. So the rib cage expands out and the diaphragm pulls down. It's like stretching out an accordion. And as we stretch, as that volume expands, as we stretch that accordion out or stretch out our thoracic cavity, we're going to see air rushes in to fill that space. Which right now, if I push this up into this little cavity, kind of compressing this volume of space inside, you can see how that compresses and collapses those lungs. But if I were to stretch it out, I don't have a lot to grab onto here, but it now has decreased the volume that's in here, and air has rushed in to fill that space. And now we've got lungs that are trying to fill with air. Let's see that in another couple of other slides. This is the last slide. I just wanted to point out that there's a structure on the lungs that we call the hilum. We're going to see this when we get into the kidneys as well. The hilum is just going to be an area where blood vessels enter, and in this case, it's also where the primary bronchi enter the lungs. So if you were to do a lung transplant or something like that, they cut it right there at the hilum, where there's the major blood vessels in the primary bronchi. Okay, jumping quickly to the next PowerPoint slide, because we're doing it all in one day here. Not too many slides. This one has mostly some physiology stuff that we talk about, and I don't have to draw a lot. It just reminds us right off the bat how the sympathetic and parasympathetic nervous system can affect these bronchial tubes. The bronchial tubes themselves are lined with muscarinic receptors. We remember that muscarinic receptors are the ones that dock acetylcholine, and when acetylcholine docks to these muscarinic receptors, it's going to cause those bronchioles to constrict. So parasympathetic nervous system is going to cause bronchial constriction. Think of rest and digest, and if you're resting and digesting, you're falling asleep on a couch, reading a book, maybe after eating a big meal. Your breath starts to become shallower and shallower, and you eventually drift off into sleep. So shallow breathing, bronchial constriction goes with this parasympathetic nervous system. But if we were in fight or flight, if we were getting hit by this sympathetic nervous system, we're going to need those bronchioles to dilate. That's going to happen because they're going to be lined with those beta-2 receptors. If you remember beta-2 receptors, we saw them on the coronary blood vessels on the surface of the heart. We saw them in skeletal muscles, and we also find them on the bronchial tubes. They're going to cause the muscles to dilate, relax. So we get more blood flow to skeletal muscles, more blood flow to the heart wall, and more air into these bronchial tubes to get to those exchange surfaces. Now to the muscles that are involved in inspiration or expiration. We'll start here with inspiration, taking in a deep breath. I mentioned the two things that happen. See if I had it in a different picture, and I guess I don't. Maybe that's the best picture that we have. The two things that happen here when you're taking in a deep breath, we can see that those external intercostals are going to cause the rib cage to come out. So external intercostals, and then the diaphragm is the other muscle. The diaphragm is going to drop down. So it's going to pull the base of the lungs with it. So the lungs, the whole thoracic cavity is being expanded, just like what was happening when I pulled this thing down, and air is rushing into that thoracic cavity to fill the space. That's how we get air into the lungs. So here's what I'll say about breathing normally. So if we are going to take in a normal breath, just like I did there, and then I released that breath, the muscles that are involved in normal breathing, there's the external intercostal and the diaphragm, that gets air in, and then if I just simply relax those same two muscles, if I relax the diaphragm and relax the external intercostals, that's sufficient to push the air out of my lungs. That's what we call a normal exhale. But down here, we want to point to muscles involved in a forceful exhale. So that would be if you're really huffing and puffing. If I wanted to force all the air out of my lungs, more than a normal exhale, so if I'm just sitting here talking and breathing, and I get to the end of one of my exhales, and I want to go and really try and force all the air out of my lungs like that. You'll notice if I really push hard, if you have your hands on your chest, when you really are forcing all that air out, you'll feel that your chest kind of sinks in a little bit. And what's doing that are those internal intercostal muscles. Those internal intercostal muscles, when you're really forcefully exhaling, they're going to compress that rib cage. That's like squeezing down on the accordion, forcing air out. In addition to the internal intercostals compressing that rib cage, you've also got the core muscles of the abdominal wall. So your rectus abdominis and your external and internal obliques, those are also helping kind of squeeze that your thoracic cavity as much as they can to force air out. So there's something in the lecture handout that wants you to know that normal inhale and normal exhale are just with external intercostal and diaphragm. If we want to get to a forceful exhale where we're really pushing all the air out, then that's when we get into internal intercostal and those core abdominal muscles that really try and force all the air out. Okay. What I'm talking about, instead of just playing the accordion, we're really referring to Boyle's law, which just shows this relationship between volume and pressure. It's an inverse relationship. So if we start to increase the volume, then by definition, the pressure is going to decrease. If we do the opposite, if we start to decrease the volume, which is what we're seeing here, as volume decreases, pressure increases. If we increase volume, we can do that, but it's going to come at the cost of pressure decreasing. Same thing that we're seeing here in the much more compressed space. So as volume decreases, pressure increases. And as volume here we've got the most volume, we've got the lowest amount of pressure. So Boyle's law is always this inverse relationship. It again is just showing how we play these lungs like accordions. This graph is showing the actual surface of the lungs. The pinkish part is the visceral pleural, and the white part is the parietal pleural, the space that the lungs have to expand into. And here's this very simplified sketch of the lungs. We've got a couple of slides to click through here, and it's just simply showing how we get to draw air into these lungs. And it's a very simplified picture of the lung. It's kind of breaking the lung down into just one alveoli, one big pocket of exchange. And you can see touching the surface of the lung is our visceral pleural. You have around the, kind of touching the inside of the rib cage is our parietal pleural. And in between there, we have that pleural cavity, that intrapleural space. Okay, so we've labeled those things down. If we go to this slide, what we've done is we've basically taken that lung and we've rounded it out so that it looks like one individual alveoli. And we know that atmospheric pressure inside that alveoli is the same as outside atmospheric pressure. If we were to break up the partial pressures of gases like we did over here in the beginning, we know that that oxygen, as it enters those lungs, it's going to end up starting to get used up. Cells are going to grab it. But pressure alone is going to be what holds these alveoli open. Notice how atmospheric pressure is at 760 millimeters of mercury. That's the same amount of pressure inside the alveoli. But just to the outside of the alveoli, there's this little space. In fact, I'll use these boxes, but I got to get closer to this. The first thing to point out is this space in here in pink that we're going to call the intraalveolar pressure. So that's inside that pocket where gas exchange is going to occur. The other thing to point out is this light blue space that's the intrapleural pressure. Notice how that intrapleural pressure is less than the intraalveolar pressure. This being higher pressure is just why this balloon wants to stay expanded and doesn't collapse. It has to be surrounded by lower pressure if it's going to stay expanded. So we want to pay attention to the difference in pressure between intrapleural pressure and intraalveolar pressure. That difference between the pressure gradient right here is what we're calling our transpulmonary pressure. Transpulmonary pressure, just between that membrane. We're looking at the difference in pressure. We will always want there to be a lower pressure around the outside of the alveoli. That's what allows that to expand. In fact, what happens if we lose that change in pressure? This shows what happens if there is what we call a traumatic pneumothorax. A traumatic pneumothorax is when you've been punctured through the chest and it actually pokes a hole through that pleural cavity. So now that intrapleural space has been exposed to outside air. And that outside air of 760 millimeters, that atmospheric pressure surrounds the alveoli. You lose that transmural pressure gradient and the alveoli collapses. It's what causes collapsed lungs. It can either have a hole from the outside, this traumatic pneumothorax, somebody stabbed you from the outside, or it can happen if you stretch the lungs too much from the inside, if there is a hole that rips open in the wall of one of these alveoli. Then you can lose your pressure from the inside. This is what we call a spontaneous pneumothorax. Okay. So either way, you want to have surrounding the outside of the lung lower pressure in that intrapleural space than you want to have in that intraalveolar space. Here this is in just a series of steps. We are starting here at the beginning before any inspiration occurs, and this is the same numbers that we started with. If I can go back to this slide, 760 millimeters of mercury outside and inside the alveoli, surrounded just in that intrapleural space by a lower pressure, 756 millimeters of mercury. What's going to change these numbers is if we change the size of the space. And that's going to happen as we start to take in air. So here we are before inspiration. All those numbers are the same numbers that we just looked at. But what starts to happen when we play this accordion of the lungs? The size of the cavity increases when we start to expand our thoracic cavity. Like we said, as the alveoli, if they're compliant and they're going to expand like a balloon, when that volume increases, the pressure inside decreases. So here we notice how this is larger than it started. This expansion of the alveoli causes the pressure inside to drop down just a little bit. And now it's below atmospheric pressure. That expansion of the alveoli and the pressure dropping inside is what allows air to rush in. So when we expand the thoracic cavity and expand those alveoli, air rushes in. And then to force that air back out, all we have to do is compress the thoracic cavity, compress those alveoli. And if we decrease the size, again, just like we saw with Boyle's law, as volume decreases, the pressure increases. And all we need to do is get that pressure just slightly above atmospheric pressure, and air is going to go the opposite direction. So during expiration, we have that compression of the thoracic cavity. There's a decrease in volume and an increase in pressure. There's a chart that shows that. We're looking at it now. It shows the blue line here is our intra-alveolar pressure, the pressure inside the alveoli. We always want this pressure higher than that pressure around the alveoli, the intrapleural pressure. Notice how, again, atmospheric pressure is the dotted line. It stays the same. As we start to inhale, what's really happening is we're expanding that alveolar space so that there's more volume so that pressure drops below atmospheric pressure. So when pressure inside the alveoli is below atmospheric pressure, air is rushing in. Then as we start to collapse the alveoli, it crosses that dotted line where now the alveoli has less volume in it. Pressure has increased above the atmospheric pressure, and as long as pressure inside the alveoli is above atmospheric pressure, then air is going to be pushed out. So we can see expiration is when the alveolar pressure is above atmospheric pressure. Inspiration is when that alveolar pressure is below atmospheric pressure. Okay. One of the last things that I want to say that's just a physiology term in here, it has to do with dead space. Dead space or dead air refers to, another way of thinking about that is spent air. This is air that's left over from a previous inhale. They're using different colors to show fresh air. They're showing fresh air in blue, and old air left over from a previous inhale, they're showing that in kind of a gray color. And all I'm trying to show here with this pulmonary ventilation, when we're getting air to and from those exchange surfaces, even if we force all the air out of those exchange surfaces, the air that's left over from the alveoli, even when we force air out, here we are forcing air out, some of this spent air that's already had the oxygen pulled out and the carbon dioxide pumped into it makes its way back into these, this would be that terminal bronchial. And then when the fresh air is pulled in at the next inhale, the first air that enters the alveoli is actually that spent air left over from a previous inhale. So every time you take in a new breath of fresh air, there's a little bit of that air that's left over from a previous inhale. So that's not going to have additional oxygen. It's going to be already latent with carbon dioxide. So that leftover spent air from a previous inhale is what we call dead space. Now we're about to graph the rest of this air as it moves to and from those exchange surfaces. This reminds us that normal pulmonary ventilation, this movement of air to and from the exchange surfaces, is about 6,000 milliliters per minute. We're going to graph this when we do our spirogram. I'll probably just pull the slides up and do it, unless we can fit it right here. We're going to see that on average, this depends on how tall you are. So you could have, sometimes, well, the textbook gives out different numbers. It typically gives larger lung capacity to males than females, but that doesn't necessarily always hold true. You could have a tall female and a short male, and typically height has more to do with lung capacity than being male or female. But when we think about average lung capacity, we can say average lung capacity is about 600 milliliters of air that you could fit into the lungs. And that air going away from those exchange surfaces and then back to the exchange surfaces is what we call our pulmonary ventilation. We're going to see that pulmonary ventilation is a product of tidal volume and respiratory rate. Respiratory rate might be obvious. That's just breaths per minute, how many times you're breathing per minute. But tidal volume, you can think of tidal volume as normal breathing. If you're just sitting there writing notes or watching TV before you go to bed, you're just doing this tidal volume, tidal breathing. The name of that comes from the tides that go in and out. That's normal movement of the tides, this tidal volume. It's normal movement of air into and out of the lungs. If I were to then like leave here and try and exercise by running up and down the steps a bunch, then I would start huffing and puffing, and I would need more air than I would do if I was just watching TV. So that gets into forceful breathing. In fact, all the stuff that I'm talking about right now is this spirogram. So I'll try and squeeze this in over here. And I'll leave that other drawing up. You can see they're using 5,700 milliliters to represent total lung volume in this individual. I'll use slightly different numbers over here. I'll draw our spirogram. Is this one better? We've got time. We'll just measure this in seconds. And over here, what we're going to measure is lung volumes. So total lung volume, that's going to be everything that we can fit into the lungs. Typically, total lung volume for, this is measured in milliliters, is typically, like I said, about 6,000. They're using 5,700 up there, and that's fine. We're going to go from 0 to 6,000 here. Somewhere in the middle is what we're going to mark off as normal breathing or tidal volume. I'll just use the same numbers there. We've got from 2,700 milliliters down to 2,200 milliliters. If I were just to section this off, I'll do it with some dotted lines. I'm going to put another mark in here at about 1,200. And this also varies. You can see they have it at 1,200 here. That's marking the residual volume. Depending on how healthy you are, this may get all the way down to like 60 or less than that. But I'll put a dotted line there too. Maybe try and stretch that a little bit differently. And then I'll just kind of cap this thing off at the top. Now let's go in here and label this thing, and I'll grab some different colors. That ought to work. Now, the thing that we're going to label right here in the middle, I was calling this tidal volume. We'll see it abbreviated just as TV. Or tidal volume. Tidal volume is going to be normal breathing. So as I graph this, what we're going to see as the line goes up, this is air going into the lungs. And the air, you can think the lungs are filling with air up to about 2,700 milliliters. That's at the end of a normal inhale, and then the line is going to come back down. This indicates that this is an exhale. And like I said, if you're just sitting there writing notes, just pay attention to your breathing for a second. You'll notice that you have air that comes in, and then you'll have air that goes back out. When you get to the end of your next exhale, you'll automatically start with your next inhale. So this normal movement of air in and out of the lungs is our tidal volume. If we start to run, like stairs or something like that, we're going to get into air that's outside of normal breathing, above and below normal breathing. So I did something earlier when I was talking. I said, I'm going to get to the end of one of my normal exhales, and then I'm just going to forcefully push out as much air as I can. So let me breathe for a second. And then at the end of one of my exhales, I'm going to just really try and, once again, force all the air out of my lungs. What I'm doing now, I've gone past a normal exhale, and I've gone down into this range down here that we're going to call our expiratory reserve volume. I'm going to abbreviate it as ERV. This ERV is expiratory reserve volume. You've got the abbreviation right here. You can think of this as past a normal exhale. And you'll notice, even if I tried to force all the air out of my lungs as I possibly can, there's still going to be a little bit of air left in there. That air that's left in the lungs, even after you've forced as much out, is called our residual volume. There's there's a certain amount of residual volume that you have to stay alive. It's much less than 1,200. I think it's somewhere around like 30. But regardless of how much residual volume it is, we can define residual volume as the amount of air that's left in the lungs even after a maximum exhale. In fact, there's actually a reflex that we will see down here called the deflation reflex, that there are sensors in your lungs, if we haven't talked about this already, that prevent you from just collapsing your lungs too much. So there's only so much I can force out of my lungs. And then there's this deflation reflex that prevents me from forcing any more out. Instead, I just have to get into breathing air into my lungs. Let me make that a little steeper there. So let's get back into this normal range of breathing, and I'll demonstrate it the other way. If I'm just doing normal breathing for a second, and then I try and do something like this and see how much air I could possibly fit into my lungs. My lungs are only so big, so if I go past a normal inhale, I'm now into this area that we're going to call the inspiratory reserve volume. We're going to abbreviate that as IRV. Inspiratory reserve volume, you can think of this is the space past a normal inhale. It takes us to the maximum amount of air that we can fit into our lungs. And this is where if you're taller and you have more lung capacity, you could fit more air into your lungs. But we still don't want to cause our lungs to expand so much that they pop like a balloon. So we're also going to see another reflex up here called the inflation reflex. If I can spell it. So we have a deflation reflex and an inflation reflex. That just prevents us from expanding our lungs to the point that they pop. That prevents us from taking in any more air and instead ensures that we're back to pushing air out of the lungs. So of all the movement of air into or out of the lungs, we have normal movement of air, this tidal volume, normal breathing. To go into the chart, we've got normal breathing here. Then we've got our past a normal inhale. This is into our inspiratory reserve volume. And depending on how big, of lungs you have, that's going to determine your total lung capacity. Obviously, the line going down, we're exhaling. If we go below that tidal volume, we're into our expiratory reserve volume. But there's still only so much air we can force out of the lungs. The amount of air that's left over in the lungs is the residual volume. So they're in your lecture handout as well as on this slide. I tried to group together some of these abbreviations. We don't see them all, but just in case you're curious, this is the entire graph. Obviously, total lung capacity is all of the amount of air that's in the lungs itself. But there's only a certain fraction of that air that we consider usable air. Normal breathing, and then forceful breathing, a forceful inhale and a forceful exhale. So this combination of tidal volume, inspiratory reserve volume, and expiratory reserve volume is what we call vital capacity. It's this little equation over here. Vital capacity is just your usable air. So it includes everything except the residual volume, the stuff that we don't use. I feel like there's one other thing. I'm going to go back to this thing real quick because also on that lecture handout, I'm seeing the key over here. There's one thing that it wants you to know, and that's this equation of pulmonary ventilation. Pulmonary ventilation is respiratory rate times however much your normal breathing is. So tidal volume times respiratory rate is the volume of air that hits those exchange surfaces every minute. Okay, I'll let those sit over there in case you want to check those things out. And I'll finish this briefly with, we've gotten through the spirogram. That leaves us just with a couple of slides talking about gas transport. Here are the two gases we've been talking about, oxygen and carbon dioxide. I said get ready because this gas transport has something to do with the red blood cell. We're going to see that oxygen, if we think of 100% of oxygen moving through the bloodstream, almost all of that oxygen is going to be bound to hemoglobin. Oops. Here is the picture of that hemoglobin molecule. At the center of that hemoglobin molecule, there's the iron group, that heme group. And that's where oxygen binds to. You can see when oxygen binds to it, it kind of changes shape to hold on to that oxygen. There are 4 of those in each hemoglobin molecule, so each hemoglobin can carry 4 of those oxygen molecules with it. What is also noteworthy is that that same binding site for oxygen, that heme site, that's the same site that carbon monoxide wants to bind to as well. The carbon monoxide and oxygen, they compete for that same binding site, that same heme site. But what's different is that carbon monoxide is much stronger. It's much more effective at binding to that site. 240 times stronger at binding to that site. So given the opportunity, carbon monoxide is going to win out every time and going to block that heme group. It's why there was the freeze last year where there were some people that had the space heaters in their house and didn't have proper ventilation, and that led to carbon monoxide poisoning. It's an odorless gas that can, like we said, cause you to suffocate without even knowing it, because what it's really doing is it's covering up this heme site, preventing oxygen from binding to it. So now your red blood cells can't deliver oxygen around the body, and the cells drowned in their own waste. So oxygen is bound to the heme site on hemoglobin. Just to go back to this slide, that's where almost all of it is carried. So if we lose that heme site, we really lose the ability to carry oxygen around the blood. Almost 2% of oxygen is going to be dissolved in the blood plasma, which we know is the watery part of the blood, not the formed elements. I think maybe this is, nope, we're in the carbon dioxide now. I thought I had another picture of oxygen. This is the only picture of oxygen transport. We can see most of that oxygen is bound to that hemoglobin, the iron site on the hemoglobin molecule. 1.5% of it is going to be dissolved in the plasma. So that takes us to carbon dioxide. And I think, just so that I can make sure that is the last slide, if we look at carbon dioxide transport, we start over here in the body cells where carbon dioxide is produced. Carbon dioxide, after being produced in these cells, jumps into the blood plasma. We're going to see that some of it, like 10% of it, is going to be dissolved in blood plasma. Then we're going to see that 30% of it, let's go back to these, 10%, 30%, and 60% for carbon dioxide. There's a couple of other ways that it can be transported as opposed to oxygen. Oxygen just has 2 pathways. Carbon dioxide has 3. We said 10% of it is going to be dissolved in the plasma, but then 30% is going to be bound to that hemoglobin molecule. That's not the majority of it. However, most of this carbon dioxide is going to be locked up in a molecule that we're going to call bicarbonate. I think we've said this before, and if not, we're going to see it again in a case study. I'm just going to use this part of the board to write something. If we haven't seen it already, we'll see it a bunch. This is what's happening in the oceans, where there's water in the oceans. If there's excess carbon dioxide that combines with water, it's going to end up forming this molecule that we're going to call carbonic acid. What makes this molecule an acid and called carbonic acid is because what an acid will do is an acid will spontaneously break apart into 2 things. One of those things that it kicks off is a bare hydrogen ion. That's what makes it an acid. What's left over, if we were just to remove a hydrogen from that, we've got this molecule, which is bicarbonate. So most of this carbon dioxide, when it gets into the bloodstream, is going to go through this process and be locked up in bicarbonate. Meaning that if there's more carbon dioxide in the bloodstream, you're going to have more of these free hydrogen ions in the bloodstream. That's going to be a case study that we have when we get into the urinary system later on. That's Unit 4 stuff. But just know there's a direct relationship with carbon dioxide in the bloodstream and how acidic your blood is. If there's more carbon dioxide in the bloodstream, there's more free hydrogen ions. Okay, I've gotten through both slides. That takes me to the lab list of things. In fact, I'll keep these PowerPoints handy in case we want to review any of those. I've got a copy of the lab list. If you didn't grab one of these, you're welcome to grab one and meet me over here near the models. Or if you want to take a break and stretch your legs for a few minutes, you're welcome to do that.